Live. Yes, madam, we are live. You can start the session now.
All right. So hello, everyone. This is our first discussion session, the live session for the course Cell Biology. And I can see some of you have joined already. So welcome to this discussion. And I think we can share some of the procedures with you. You can actually type your questions in the chat box if you've joined on Zoom, and you can also type questions in the YouTube if you're joined in by YouTube. All right, so yes, of course, I'm Professor Shikha Laloria, and I hope you all can see all of us. The two TAs are also here. They can introduce themselves. Devalina Dasgupta and Swagatinath Gopinath, they are also with us. So please introduce yourselves momentarily.
Hi guys, hi everyone. This is Devolina. I think you guys know my name from the discussion forum. We have been trying our best to answer your questions. So hope you guys are well, doing well, and hope you ask your all the queries here. Ma'am is here, and we are here to solve your doubt.
Hello everyone, this is Swagatnath, and I hope you guys recognize me from the discussion forum. And here we are trying to resolve your queries, any queries related to the subject, and you can type the questions along the YouTube chat box. We are there, and we will try to communicate with ourselves, and we try to resolve the doubt. So hope this session goes fruitful.
Over to you, ma'am.
All right, so I think you all can also type the questions in the chat box in Zoom, because I don't have the YouTube available exactly right now, but I'll try to connect to it as the session goes on. So we had posted this Google Sheet, and I think some of you are typing some questions there. So I thought I'll try to resolve it. And any other questions, you can also type in the chat box. So so let's start. I think one of the questions many of the students had pertains to the mitotic exit network. Now, that is a rather complicated subject, and I'm glad those who had the doubts have sort of gone ahead and asked the question. And let's try to discuss that. It's a very interesting topic. And the lectures pertaining to that are my lecture on mitosis, the second part, as well as it has also been touched upon in the lecture on cytokinesis by Dr. Palani. Okay, so I'll try to synthesize both of those, and also I've tried to update my knowledge also, because as you know, research, it keeps progressing, and there are some new interesting points also that came up when I was trying to look at the answer. So let's see. I think I would like to share my screen. So let's just do that. 1 minute. Just 1 moment. Actually, maybe I want to exit this. Yeah, I'll just turn on the slideshow actually, because that's more easy for all of you to see. But then Wait a minute. All right. So I'm sharing my screen. And let me know when you all can see that.
Yes, ma'am.
Is it visible?
No, not
No,
till now.
not yet.
Okay. How about now?
Yeah, now
Yeah.
it's showing.
Okay. So I'll also just touch upon some of the points I think when I was preparing for this, thinking about what to say, we can maybe enter this mode. Right. You all, of course, have this information. Yeah, one thing I want to say to all the students is you do keep checking the discussion forum for any further updates and announcements, and also check the ask a question section because I post certain PDFs there on and off, the relevant transcripts and so on. And you all can go through this. Many of you ask questions to us about the exams, and I think it has been quite clearly mentioned in this announcement, which is also on the forum. So yeah, coming to the mitotic exit network. So I think, let me just say, the student who asked this question, I'll just like to mention the name also. There are a few questions. Deblina, could you look that up? There are, yeah. I think Rajesh, Chetan Rajesh Lachure had this question. And also Tejas had some question about the mitotic exit network. Also Charukesh Kannan, all three of you. So this is a very popular question. And I hope that, we can shed some light on it. So am I out of the slideshow now?
No, ma'am, the slides are still showing.
No, slideshow. You are out of slideshow. No.
Okay, yeah, now we are back. Right, so this is a very interesting question, pertaining to regulation in cell biology. And I actually like to read about this myself also. So this is a very nice example of spatial regulation of the cell cycle. And so the mitotic exit network, as you know, it's a signaling pathway, which is towards the end of mitosis. And after this, the cells, they enter and execute cytokinesis. So how does the cell know when to do that? Okay, that's what it's about. And, just to distill the real question, how is spatial information in the cell translated into a chemical signal? That is what, as a scientist, we would like to understand. So it involves two parts. I mean, the mitotic exit network, it is regulated by the spindle position checkpoint. Okay, we've discussed checkpoints in detail in the class, as you know. So what this spindle position checkpoint does is it senses the spindle position. And so within the cell, so in budding yeast, you have got the mother cell and you have the daughter bud coming out. And the spindle, it's dynamic, so it could be moving around. And it's in the mother cell and it can keep changing its position, right? And actually, for proper cell division to happen, the chromosomes, they have to be segregated, and one should be in the mother cell, one set, and the other should be in the daughter cell. Only then the two progeny cells, they will get the equal number of chromosomes. So if the cell division happens when the set of chromosomes they are not in the daughter cell, then you'll get one cell without chromosomes, and it would be dead. So it's a very important checkpoint. So as long as the spindle is not properly oriented, as long as it's misaligned, then this checkpoint is active and it inhibits the signaling pathway, the mitotic exit network. Okay, and so there are different models about it, and one of them I did discuss, or one part of it I have discussed in the class in the earlier lecture, And there are also some new angles to it. So the spindle position is sensed by MEN inhibitory and activating zones. Okay. And there is a sensor also that changes its position, can change its position within. So the inhibitory zone is located in the mother cell, and the activating zone, the mitotic exit activating zone, is located in the bud cell. And the spindle pole, which is present at the two ends of the spindle, that is where the components of this mitotic exit network are present. It functions as the sensor, and its position can change because there are microtubules and motor proteins working in the cell also. And so it keeps on sort of changing its position. And it can happen that one of the spindle pole bodies will enter into the daughter bud. If it comes to that region where the connection between the mother cell and the daughter cell, if it passes through that and it enters into the daughter cell or the daughter bud, that is when it actually escapes. The spindle pole body is no longer now under the influence of the inhibitory zone, which is present in the mother cell. And one of the important inhibitors is KIN4, which is a kinase, which is present in the mother cell. And when it enters into the daughter cell, then there is an activating molecule LTE1, which is only present in the daughter cell. So when it enters there, it comes into proximity or vicinity of LTE1, which then activates TEM1, okay, which is an important protein. It's a key molecule at the start of this pathway. So yes, the mitotic exit network can be activated when the spindle is properly oriented and one of the spindle pole bodies has actually entered into the daughter bud, and one of them remains in the mother. So it's a signaling cascade, and then when it's activated, it drives the cells out of mitosis by activating CDC14, which is a phosphatase, and this protein, of course, was discovered from the screen, the CDC mutant screen that we have discussed in the course. Okay, there are two important references here, and I think maybe I can use a pointer, yes. So these two references, they're actually quite interesting for the students who were very interested in knowing about this topic. You will learn much more, and these are just some points to sort of remind me what I must say. So I'm not really going to read all of it, but in fact, I've already explained that. So one of the important proteins is this GTPase TEM1, which we have mentioned in the class. It's present on the spindle pole bodies. And this TEM1, it can be in a GTP bound form or a GDP bound form. And when it's activated, it actually signals to certain kinase molecules, which are also present close to it. And then it can result in the activation of CDC14. In fact, CDC14 is normally present in the nucleolus. So when this pathway is activated, CDC14 will be released from the nucleolus. It will spread out into the nucleus. It will spread into the cytoplasm. And being a phosphatase, it goes and dephosphorates many of the proteins that were earlier being phosphorylated by the mitotic CDK. So it reverses the effect of the mCDK. It counteracts mCDK, which was letting the cells be in mitosis and has activated mitosis. So, so you all know that now, and TEM1, it is positively regulated by LTE1. So the important thing about LTE1 is its spatial regulation. That is, it is present only in the daughter bud. Okay, so therefore only when the one spindle pole body enters there, then TEM1, which is present at the spindle pole body, it comes in contact with LTE1. So LTE1 is a exchange factor. Okay, So it can cause exchange of the bound nucleotide GTP. So there are two forms, GTP bound and GDP bound TEM1. And there's also a gap. GTP is activating protein that converts TEM1 from the GTP bound form to the GDP bound form. And LT1 can cause the exchange of GDP for GTP, so it can convert it into the GTP bound form. Okay, so so this is the gap is the BUP2 and BFA complex, and there's additional regulation which we have not discussed earlier, but there's another kinase CDC5, the polo-like kinase, which inhibits this gap complex. Okay. So how does it work? The spindle pole position checkpoint, it inhibits the mitotic exit network at the level of TEM1 activation. So that's the key event, the key molecule, and the key event is TEM1 activation. And so there are some other proteins I mentioned, KIN4 and its regulator, protein phosphatase. Let's not focus on that, but KIN4 is sort of interesting and important. It's present in the mother cell cortex throughout the cell cycle. So like LT1, this also has an asymmetrical distribution or interesting spatial distribution. It's not present in both the cells. In the emerging daughter bud, it's not present, but it's there in the mother. Okay, and it keeps the gap active. And therefore, when the gap is active, then TEM1 is in the GDP bound inactive state. So as long as it's there in the spindle pole body in the mother cell, will have this protein inactive. And therefore, the pathway is not activated. So again, when the spindle pole body enters into the bud, there's LTE1. So it's no longer under the negative influence of KIN4. And also it comes in contact with the LTE1. And then it becomes active, and then it can sort of trigger the mitotic exit network. So some researchers, I think from Angelica Amon's lab and some other labs also, they have discussed this zone model, which is quite interesting. So they proposed this model. And according to this model, the TEM1 bearing spindle pole body, it should first escape the zone of negative influence or the zone of inhibition in the mother cell. And then it escapes that and it enters the zone of activation in the daughter cell where LT1 is present. And that is shown in this graphic here. It's quite simple, actually. So you all can see the full slide, right? Or is a part of it missing? Because I can see panel on this.
Visualize the slide.
Only the slide, right?
Yeah.
Okay, because I can see something else on the side. All right. So, yeah. So in this, it's actually quite simply shown. The red over here is they're showing the distribution of KIN4, and here this is the daughter bud, and there you can see only LT1 is only present there. Okay, and the first part here, the spindle is misaligned. So these are the two spindle poles, the red balls. Okay, and in this figure, they are both of them are in the mother cell. So the spindle is not really oriented properly yet it can be moving around. So while in this state, then TEM1 will be, it is not active. Okay, as I explained, but when it comes in this orientation where now one of the spindle pole bodies has entered into the daughter, then that's where LT1 is present, and that's where the blue symbol here indicates that it's activated now. Okay, because of the GTP exchange activity of LTE1. So it's now present in the GTP bound active form. And so another way, so this is the general pathway. There are two checkpoints, the spindle assembly checkpoint and the spindle position checkpoint. And these are the regulators. So here it's just shown in a more graphical form. So these are the key molecules. The TEM1 GTP, it's the active form, and this is the inactive form, right? And the gap converts the TEM GTP into the GDP. And LT, which is only there in the daughter cell, it converts it into the active form. And after that happens, then again, there are some complex signaling cascades, okay, which I don't know the details of, and it's a evolving field, but they know some of the components such as CDC15 and some of these other proteins mentioned here. So they bring about the release of CDC14 from the nucleolus and its activation. So there is actually this NET1 and some other proteins. They form a complex and they restrain CDC14 or sequester it within the nucleolus. And then, so this is released, it comes out, and then it has all these different activities that we also mentioned in the class. So it can actually dephosphorate the APC activator CDH1. Okay, and also SIC1, which is the CDK inhibitor. And there's lowered CDK activity, and this causes the exit from mitosis. So that's a thing. And this is an interesting graphic showing everything that I've explained. This is from an interesting review by Andy Hoyt. And I think we have shared that reference also, but you all can look into it. So here it is shown LTE1, the yellow, its distribution, the GEF is mentioned here. And again, when this pole has entered, here you have the GTP bound form. And this is when the spindle orientation is just right. The green dots are signifying CDC14. You can see how it was sequestered here in the nucleolus when the spindle was misoriented, and when it's properly oriented, it's released. And then because it's a phosphatase, it can diffuse and go and find its targets, and effectively it counteracts the activity of the MCDK. Okay, so I hope that's clear. This is the graphic that I shared in the class, and basically trying to explain, yeah, so there is some complicated 2 kinase cascades over here, which ultimately they bring about this release of CDC14. And I think I already showed the nicer version of this earlier. Okay, so what does CDC14 do? It actually dephosphorates MCDK substrates and also SIC1 and CDH1. And it also has some role in cytokinesis, as Dr. Palani had explained. Now, this is his slide, and I wanted to use it to show you that indeed this happens. So here he's tagged CDC14 with the green fluorescent protein. Okay, so by fluorescence microscopy, now we can follow CDC14. It gives off green fluorescence under UV. And this is a time course experiment. So it is showing how when the orientation is proper, you can see the signal changing. So here it is sort of concentrated dark green signal. But in the following time points, you can see now more diffuse signal, and it is spread out in the cell body, the green signal, which implies that, yes, CDC14 is released into the rest of the cell. And you can see that progressing over here. Now you can see the cytokinesis happening also. And then again, it gets concentrated into the nucleolus. So I hope that part is clear now. Okay. Maybe I'll stop screen sharing, and I think I would like to see what else is going on in the Zoom. I'm not. Okay. Am I back here, or there seems to be some problem in returning back to the Zoom?
You are visible now. with screen share.
I'm not able to. Okay, maybe I stopped the share. Yes. All right, I'm back. So students are there. Let me just see. Abhishek is here. Good afternoon, Abhishek. So any more questions? Let me just check that out. So how am I doing on time? Yeah, already 25 minutes. So okay, Tejas also had a question. He's asking what happens if there's a mutation in the gene which codes for any of the components of the mitotic exit network. For example, CDC 15 or 14. I can't actually read the full thing. Yeah, so that's a good question. And, we are very strong on genetics, that tells us, in fact, many of these pathways, they were solved in this way by looking at mutants. And then, in fact, many of the proteins were discovered that way. So if you recall, we discussed the CDC mutant screen from the Hartwell lab that was done in budding yeast. And in that, many different CDC mutants were found, and CDC 14 and 15 were also among them. Okay. And so if you have a mutation in CDC 14 or 15, you get a cell cycle arrest. And it's a sort of a telophase arrest where the chromosomes have segregated. The spindle pole bodies are one in the mother cell, one is in the daughter cell. And also, it has undergone anaphase B. So the spindle has elongated, and then they are sort of present at the two ends, right? But cytokinesis has not yet occurred. So there's a characteristic arrest phenotype of the CDC 14 and 15 mutants, which is very much characteristic of a telophase or a late telophase type of arrest. All right, so it basically signifies that the cell cannot go forward and undergo cytokinesis. There is some defect, and which is very satisfying, because now we know that in fact these proteins, they are part of this signaling network, which is important for cytokinesis. Okay, now what else is there? Anybody else? Somebody else has joined. Arokia Paul Raj. Hi. And I don't know if there are any questions coming on YouTube. Maybe TS can prompt me, or just give me a minute. I'll try to connect myself.
No.
So actually, there was some question I remember regarding the primary and secondary screen. Maybe Debalina, you can take a moment to explain that, okay, to all the students. Yeah, are you there, Debalina? Am I audible? Oh, yeah, you guys are here.
Hi, ma'am. Hello.
Yeah, one of you, I think it'll be useful for students to know about the primary and secondary screen. You can take the example of the CDC mutant screen or the PDS mutant screen, any of those. I think Debalina had explained it also in the forum quite nicely. Is she there?
Yes.
Yeah.
She's here.
Do you want to explain? Please go ahead. I'm just trying to look at the YouTube right now.
Give me a moment, ma'am, please.
A moment, please, ma'am.
So basically, we do screens to find mutants. Very often you'll be studying some process that interests you. In case of the Hartwell lab, they were interested in looking at cell division, the cell cycle. So they had designed the screen, so they can pull out mutants that might be defective in that. And so while planning a screen, you would want to, there'll be lots of mutants and you have to screen thousands of mutants. So it's a lot of work. So you want to first enrich, you have to put a primary screening step to sort of enrich for the possibility of getting mutants of the type that you want. And then among that subset, you want to go ahead and then examine them. And that step may be a little more laborious, involving microscopy. So you want to work with a smaller number there. So those are the thoughts behind that. Okay, so I guess we should continue because I didn't hear back. Yeah, so in the case of the CDC mutant screen, they did a screen for temperature sensitive mutants. Everybody's clear on that, right? They are conditional mutants, temperature sensitive mutants. And the reasoning behind that was that, right. So the reasoning behind that was that, mutants which are defective in cell cycle, they are likely to be there in essential genes, genes which are essential for viability for the cell to divide. So you cannot just inactivate that gene and expect to study its function. So you need a conditional mutant. And the type they chose was TS, or temperature sensitive mutants. So they made a lot of mutants. They added a mutagen to the cells and then screened all the resulting mutants for temperature sensitivity and made a big collection of TS mutants. And so that is the primary screen, okay, where you get a large number. And then the secondary screen in their case was they were looking for cell division cycle mutants. So they visualized those mutants at the restrictive or non-permissive temperature under the microscope and looked at the budding morphology, because in case of budding is cell cycle, that's the neat thing. Just by looking at the cell, the size of the mother and the daughter cells, and particularly the size of the daughter bud, you can get a good idea in which stage of the cell cycle it might be. So they examined microscopically all those mutants at the restrictive temperature and looked for mutants where there were clear-cut cell cycle arrests or some changes in the morphology also, in case there were some elongated buds and so on. And then took those, and those were called the CDC mutants because they appeared to be defective in cell division cycle. And then they went ahead and further characterized them. So, okay. So that's the primary and the secondary screen. Okay, student of radiotherapy, Unaini Joshi. So cells after irradiation, cells will activate the DNA damage checkpoint pathway, and it basically prevents the cell cycle from progressing forward till it gives the cell time to repair the damage. Okay. And I think this will be covered in one of the future lectures. We have not yet come to this point. So the DNA damage checkpoint will sense that there is damage in the DNA, in this case induced by radiation. And again, there's a signaling cascade that prevents it prevents a cell cycle progression. And what it's doing is giving the cell time to recover or repair the damage. So it will try to do that. And then there is another option when there is too much damage and the cell is not able to repair it. Then there's also the option of cell death. So in that case, the cell can enter into the apoptotic cell death pathway. So those are the two responses. And if you wait and look at some of the future lectures coming up, I think we have explained this also. Okay, so centrosome, of course, it's the same, the function would be the same. So what we are referring to as the spindle pole body in more complex organisms, it's referred to as the centrosome. Okay, Abhishek, that's your question. I hope it's clear now. So I will maybe in the meantime go back. So I am not able to check the YouTube over here. So I hope the TAs are doing that and checking the questions. And you can type.
Yeah, I am checking the YouTube. So far, no questions from YouTube.
Okay.
I'm checking.
Okay, if there are any, you can type them. And also I want to tell all the students, I'll try my best to resolve as many questions as possible today. And if any is remaining, you all know we also have the discussion forum. So don't worry if your question was not answered by chance, we will still address that in the discussion forum. Okay. So in the meantime, I will like to address one of the other questions also, which came on our the form that we have posted for this purpose. And just one moment. Let me just check it out. I think I'm not sharing screen right now, right? Okay, yeah. So Charukesh Kannan also had a question regarding, I guess this was an assignment question. So it's a bit confused about this, that wants to know that in which stage the licensing of replication occurs. It's a bit confusing because, you all know replication occurs in S phase. So what's the question is, why do we think it's in MG1? Shouldn't it be in S phase? It deals with replication. So I'll try to actually explain this. And this is also a somewhat complex topic. I'm trying to still see the YouTube if I can, but yeah. So let's talk about that, about licensing of DNA replication origins. That's a very important key event. And some of you, of course, have doubts about that. So I'll try to address that one now. Okay. So you all know, obviously, during cell division, the DNA, it should be duplicated very precisely, and you don't want polyploidy occurring. So there is a regulatory mechanism in the cell which ensures that DNA replication in normal cells occurs only once per cell cycle. And the licensing of replication origins is a very important step to ensure that. Okay, and the molecules that are important for this, of course, the CDKs, they are master regulators of the cell cycle, and the initiator proteins in DNA replication, such as ORC, the origin recognition complex, and some regulators such as CDC6, CDT1, and also the MCM complex. So the MCM is a helicase. And that's a key molecule. If it's bound to the origin, then the origin is licensed. If it's not bound, then that origin cannot be activated. And so it's no longer licensed. That's the key thing that you need to remember. Okay, so when this MCM complex is loaded on the chromatin, then that is when you will formally define the replication origin as being licensed for replication. Now, what does licensing mean? It means it's a kind of a permission, like, okay, now you're ready, you can go ahead when the right time arises, right? We have a license to drive, we can take that and we are allowed to drive when the need arises. So in the same way, when MCM is bound to the origin, it's ready, it's poised for replicating. It can when the opportunity arises. And what is the opportunity? It is the proper signaling which tells the cell that conditions are right to divide. And that's when the cells, they cross that restriction point or the commitment point in the cell cycle that we discussed many times. So I hope you all understood that part, that there is signaling which happens, which actually triggers the initiation of replication. And replication can start only if MCM proteins were present there and that pre-replicative complex is properly assembled. Okay, so I'll just review that perhaps real quickly over here. I think once again, I have to share the screen. All right. So from the class, I don't want to actually see this thumbnail. Okay. You can see my screen now, all of you?
Yes, ma'am.
Okay, great. So yeah, so there are two things to remember. Licensing of origins. For initiation of replication, there are two main things. One is the licensing. That happens in late M and early G1, all right? So that is the correct answer. And what really happens is, as I told you, the replicative helicases, they bind at the origins and they form the pre-RC. And this is the key event. And initiation can occur only when these origins have the pre-RC assembled. Okay. And then in S phase, so after it has to cross from G1 to S, that important commitment point, the helicase will be activated, and then there's DNA unwinding and initiation of DNA synthesis, and the helicase actually moves out of the origin along with all those other replication factors. So the original site where it was bound doesn't have the helicase bound there anymore. And therefore it has become unlicensed, right? And then DNA replication proceeds and it gets completed. So yeah, the origin site now lacks the bound helicase. It can't reinitiate replication until it binds there again, which only happens after the M phase in the next G1. Okay, and this ensures that the origins, they are activated only once per cell cycle. Okay. Right, so this is the key step, and this you would remember from the lecture that I gave in class. What's going on here? This is the licensing step. It happens in late M, early G1. And here's the complex on the DNA org. The origin recognition complex is bound to these sites throughout the cell cycle, okay? And it serves as a landing pad for other proteins which regulate the initiation. One of them is CDC6. It's present at low levels, but its level increases in G1 transiently, and which is why this process is happening in G1. Okay, so it binds org, and then this is important for recruiting the MCM helicase. So CDC6 will recruit the MCM helicase, and then there's another protein CDT1, which also helps in loading it on the origin. And this is the complex where two numbers of this MCM complex are there and the ORC is there at the origin, that is the pre-replicative complex. So it needs to be there. And only after that will DNA replication happens when the proper signal is perceived. Okay, and we talked about this. So you can see when DNA replication is triggered, now this is the origin site. And I'm actually not in the view, side view, but you all can see that. Maybe we should do that, yes. Okay, so you can see this. So ORC is there, this is where the origin was. But because the replication was triggered, now the helicase, it's moving with the fork and the other replication factors. So it clears out of this region. So now this origin, now it's been replicated, this region, it's double-stranded. It's not going to fire again because it doesn't have that configuration. It's not licensed anymore. Okay, and this is the summary of it, and we have discussed this in class. So what really happens is that after when the activation of the SCDK happens, you get origin firing, and all the replication proteins and MCM is activated. But SCDK also phosphorylates ORC, and also it causes CDC6 to dissociate from the origin. Okay, and so CDC6 is phosphorylated and, excuse me, it's ubiculated by SCF and E3 ligase. And it gets degraded. And likewise, the MCM proteins are phosphorylated and they are exported from the nucleus. So now they are no longer there also in the nucleus to bind and reassemble at the origin. Okay. And of course, SCDK activity is high, and then that is followed by MCDK, which can also phosphorylate CDC6 and MCM. So even when mitosis is going on, CDC6 and MCM, they are in the phosphorylated form and one of them is getting degraded, the other is exiting, it's no longer in the nucleus, and therefore re-replication is prevented. But at the end of mitosis, when CDK activity reduces, we just talked about the mitotic exit network, right? So that's what's happening at the end. CDK activity reduces, and then the dephosphorylated forms of CDC6 and MCM would accumulate, and in that form, they can actually bind again at the origin, and you can get proper pre-RC assembly. And now the origin can get licensed again. That is after M, at the end of M phase. And it's ready for another round of DNA replication. And just to remind you, it doesn't happen all the time. So this is G1. Cell has to cross this restriction point. It makes a commitment to enter into the S phase. And that's done by signaling pathways, of course. When you have these growth factor signal, and this results, we've discussed the details of that, how D-type cyclin is synthesized, and you have the active G1 CDK, which causes the cell to cross this restriction point and initiate the DNA replication. Okay. All right. I'll just take a moment. Right. So I also noticed there was a question about Xenopus eggs, why they are useful and so on. So those cells, they have been used in studies in mitosis, as you know. So they were very useful to study because those cells, they are primed for cell division. They store large number of macromolecules which are required for cell division, and basically they are waiting for after fertilization happens, then you would have a series of divisions, and the first series of divisions, there is no growth phase actually. So whatever components are stored in those cells, they are useful. So the cell has stored up, it's waiting basically for the next step. So, they can be used to prepare extracts and carry out in vitro studies, also in vitro studies on cell division. And another very important feature of those cells is they are pretty big. I mean, you can see them with the naked eye. It could be about a millimeter in size. And so certainly under even low magnification microscope, you can see it quite readily. And because of that, it was possible to do certain manipulations on them, such as the experiment where MPF was discovered, the micro injection experiment. So because of the large cell size, it was possible with the help of capillary sort of needle to inject test substances into those cells, or extracts, as was done in those experiments that we discussed in the class. So that was one of the important features of Xenopus eggs and why they are such a favorite model in cell cycle studies. And also, you can take the extracts, and if you add the sperm nucleus, you can actually see in vitro spindle formation, and a lot of interesting experiments can be done using the extracts from those cells also. I'm trying to look at some of the other questions as well, the scientific questions. Some of them require really detailed answers, and I think TAs are doing a pretty good job answering some of those questions. So is there anything happening, any questions coming on the YouTube channel?
Hi, ma'am.
Yeah, do save them also, because I think we are kind of running out of time. So I'm asking the TAs to please save those questions.
Sure, ma'am. We'll clear the question.
Right. If there are any, then you can either post them here or definitely save the questions also.
Sure, ma'am.
Yeah. Was there any other question either of you noticed in the discussion forum, or something which needs to be discussed here?
Somebody has asked to explain lamine filament. I think it's Vinita S.
Yeah, could you explain that? I missed that. Let me see.
It is there in between.
Yeah. Isn't that in some of the later lectures?
I think.
Yeah, it will be.
Yeah, it will be there in the coming lectures.
Right. So we can discuss this next time. But yeah, these are interesting. Just below the nuclear envelope, you have lamin filaments, and they form sort of a meshwork below the nuclear envelope. And it's actually quite important in regulation also, because there are some diseases, aging related diseases, progeria, where you have defects in that. And that will be coming in an upcoming lecture, Vinita. So you can please check it and we can discuss it next time. Cytokinesis. This is the lecture by Dr. Palani, right? So the contractile ring is, in case of budding yeast, you actually see that. And he has explained very nicely. I would suggest you to go and view his lecture. I think it's already run in the 4th week. So you're asking, there are a lot of details in that. That's basically the mitotic exit network. It is activated and then it triggers the process of cytokinesis. And I think I cannot go into the details of that right now, but there is a detailed lecture on that. So I think you can look that up, Chetan, right?
So we are done, madam.
Yeah. I think, yeah, it's almost time, right? I think
Yeah.
we can end it. So thanks, students, for coming. And I would again remind the TAs to save the questions. But I think we addressed most of them. And the people who want details, they should actually revisit those lectures. And if there's any doubt, then next time, definitely we will address it, because we are planning to have at least one more session in this course. So save your doubts for that. And I hope you are enjoying the lectures and trying to solve the assignments to the best of your ability. And do try to understand and look up the references also. In some of the lectures, there are some references cited. And today also I mentioned 2 references. I think those are interesting to read. And I would encourage all of you actually to read those, to understand the regulation of the mitotic exit network. Okay. Thank you all. All the best.
Okay, madam. Thank you.
Thank you, everyone.
So I'll end the session in YouTube now.
Yeah, please go ahead.
YouTube live has been